to USB drive. This is it?
Yeah, that's it.
So we will. You can, if you want to, you can look at it up there.
It is big.
Go ahead.
Yeah, it tacked on. All right, looks good to me. Cool. I guess I can cycle through. Make sure that we'll have a few animations to even put our pictures up there. Who we are. Update on the budget. And all the stuff. Grant types. Yeah, it's gonna be fine. I'll put it. Yeah, that's good.
Sweet. All right.
That's good. I'll take them. Yeah,
Yeah, go ahead. You should be fine. You want me to
look at it real quick.
do you want me to eject it, or do you care?
Yeah, just eject it. You'll probably be alright.
Yep. Good.
Good. Thanks. I'm taking liberties here.
Editing it?
I have to put my email address at the end. Just take a second.
We're gonna get you a new name tag, Doug Thompson. I don't know if that would be on it. Here's the spell his name.
And it's not Kevin. Yeah, it's alright.
You opened this up from that folder that they're all in?
Yeah, I found it through it.
Great. As long as we got the right
Hang
one.
my way through.
Alright, we should.
It'll take a while to save that.
This is you?
Yeah, and it has a ton of stuff on it.
Oh, there you go.
Oh,
Yeah, we can open it up and check it, but otherwise I'm going to drag it to a different. Yep. I don't know if you want to run through it at all.
yeah.
That won't be up there. That's part of we're streaming it, so they'll be able to see the presentation and you as all this view right here.
Oh, really? Huh.
Yeah. And then we also have a clicker for it too.
All right. Fine.
Cool.
And there's going to be a pointer.
Yep, right here. It already works. And you can go through the slides too.
Oh, okay.
Hold on. I'll eject so it's off. This keeps telling me it's in use. That's not true.
Keeps telling you what?
Oh, that's right. That's not the one. Eject. I just don't want to corrupt anything for you. Come on. If you're okay with it, you can also.
I'm just going to take it
Oh,
out.
yeah,
I
just
do it all the time.
Yeah, it's been ejected. Yeah. Cool. Good?
Yeah, good. Thank you.
Upstairs.
Good morning. Thank you for joining us this early in the morning. It's great to see you all here. First of all, there were a few questions about Wi-Fi. You can log on to the guest server. And Brent, I think he just ran out of the room, but he'll be running all the AV, so if you can't log on to that, flag him down and he'll help you out. Again, welcome. This is our second annual symposium. So Crop Science Department, DuPont Pioneer, and the Graduate Planning Committee welcome you. And we're really excited for this great lineup of talks, and we're excited to host you here today. So Brian is going to introduce our speaker for our opening remarks, and enjoy the conference.
So it's my pleasure to introduce Tabaré Abadi de Leon. He is the lead of academic outreach for Pioneer and a good friend of mine, and honestly the reason this whole symposium series is possible, he'll go into what it's all about, all the different universities that are a part of it, but it's his vision that's possible. So without further ado, welcome up Tabaré. Here's this will
okay.
two slides.
Okay. Thank you.
You want to try this?
Or I can do here. I mean,
That.
yeah.
It's up to you. If you want to walk around, though, you should use.
No. I just not too long. Don't worry. I'm not gonna bore you guys too much. Thank you guys, and thank you, Brian. I also feel I am a very good friend of yours. So good to be here. Thank you to the organizers for setting this up, and for a second year. It is an honor for us to sponsor your event and to work together along the year to put an event like this together. What I would like to do on this introduction is to introduce the audience to what are we trying to do by sponsoring these individual events around the world, and how do we bring them together in what we call the DuPont Plant Science Symposia series. Okay, and so just a little bit of an introduction on that. The objectives or the general perspective that we have with this program is to empower grad students to drive their own educational agenda. Of course, this is not 100% of their educational agenda, but it's an opportunity for them to exercise freedom to actually make decisions in an academic setting that hopefully will empower them to expand their possibilities of making their own choices in other areas of their own program too. So the objectives of the program are to contribute to education, and especially on the areas related of expanding entrepreneurial, communication, risk management, etc. skills that of course are very, very important for any career in science or actually in any other field. The other objective is to do outreach. So it's an opportunity for the students to actually open up an event to society and to communicate their ideas, and by inviting people into an open forum in which ideas are presented and discussed. And also to expose or to present future graduates to different career opportunities, right? I mean, people that are coming have had different options in their careers, and we hope that this could be inspiring for the students to actually begin to think what would be their future careers when they graduate. So how all these events work? Well, all these events are fully run by students. We provide support and encouragement and whatever else that we do. But the important thing is we want students to actually feel completely free on running these events. They are the real owners of the event. And again, this is mainly designed to be an educational experience, so there are other components that might be very important at the local level, but in the larger picture, what we really emphasize is which are the skills that the students organizing these events acquire by running them. Students select topics and speakers. We do not give any guideline in that area. you know, and we are willing to take any challenge that the students would like to raise to us on this. And we understand that the science that is presented is relevant. And by relevant, it means that, you know, the students might have had a good discussion on what they were going to be talking about and whom they will be inviting, and hopefully they will benefit by the interaction generated by the meeting. And in most of the cases, and it's certainly so in this case, the participation is 100% free, and not only in presence but also online. So again, it's a pretty open forum at a local level that we try to generate and build connections globally. So I want to present with you with a little bit of some of the dimensions, you know, that we want to achieve with this forum. And one is, I would say, the global dimension of the forum. And so, and to do so, I will present you with the story of how we got to the present. And we started in 2008 with a partnership with the University of Minnesota. Rex Bernardo sent us a note asking us to support a student-run event at the University of Minnesota. We provided support and participated on the organization of the 3 first events in Minnesota, and they were extremely successful. Every year, the audience increased and increased, and it, you know, their own brand. I lost something here. Am I there? Okay, their own brand expanded and grew, and so it was a great idea. Let me see if I can get it back here. I think I got completely off here, but I am good at talking without any help. So we had a very successful year, 3 years there, and at the end of the 3rd year, we realized that this was an excellent idea. So why don't take this to other places, right? So in 2011, we invited the students of the University of Minnesota to help us promote the same idea and the same kind of forum in Wisconsin. And we did it in Wisconsin, and it worked very well. There was a very good partnership between the students of the 2 universities, and we began to move along, right? And so we went to Wisconsin, and then we kept expanding. And I don't have the slide there, but I do have. You might have seen the flyer, and in the flyer you have all the universities that belong to the series at this point. And so we expanded and expanded and expanded every year. And so this last year we added 15 new universities and centers into the series, spanning all the continents. I would say all the, you know, fully inhabited, you know, agriculturally feasible continents. And this year we are going to have an event. The last event of the year is going to be in Queensland, Australia. And so by the end of the year, we would have reached more than 90 events. And you know, our expectation is that we would have had more than 15,000 participants around the world. And so historically on the event. So, you know, I mean, it is a significant contribution to the expansion of ideas in science and possibilities for people to advance their education. So another dimension that, of course, I won't be able to show you here, but I had it in my slide, was what are the connections that have been developed during this process. And one of the major connections has been the possibility of having people travel around the world as speakers in this event. And although most of these events are very regional, we have had significant contributions of traveling and exchange of scientists around the world in this. And the last dimension that I wanted to mention here is the student participation on the governance of the series. Every year we try to have a gathering of the organizers of the different events in different universities to come together and make decisions on which are the key things that are important to keep the series together. And of course, one of those things is, communication. And we have been, as a group, we have decided how communication is going to happen. I mean, and one of the major things is each university has their own web page, but we do have a centralized web page in which we gather all the links to the individual events. Okay, and so if we were to have the slides, or maybe we might. Computer science is a very important component in life today, and one has to be very aware of that. So, very rapidly, these are all the events that we are having. This is this year, and as you see, we expanded, greatly around the world. I'm not going to get out from here because it might disappear. These are all the connections that were generated over the years, and as you see, there's a big emphasis on regional connections, but we also have, connections across continents. And these are all the speakers and connections that were generated during the 10 years of the series in North America. And voila, these are some of the speakers and key speakers that have participated on the series along the years. So one of the things that we have been doing greatly is to try to gather as much data as possible on this, because we think that at this point we have a very, very nice sample of what is the demand in this area in different parts of the world. And we think that it's very interesting. I mean, I hope that some of you would like to be part of this light sometime, and we would like that too. And these are the major topics covered by the series. As you see, breeding is the fundamental topic in which we started, but we have been expanding to a lot of other areas, and we are really willing to expand even further as the definition of breeding and in general, the areas of interest for agriculture expand rapidly. Again, student participation is very important. Especially on participating on the governance. I mentioned the component related to the web pages, but another thing that I didn't mention is one of the things that came out from the decisions made with students was the idea of supporting travel grants for students to travel between universities and participate on the events on other universities. And these are some of the connections that were generated during this last year on the travel grants that were offered to students in North America. And as you see, it has been significant, and of course it does include Mexico, because hey, Mexico is also part of North America, but there have been very, very good connections there, and as you see, we also have events in Canada too. And again, each event has their own web page, and all of them, but we do have a centralized web page, and in that centralized web page is a very, very easy web page to go. We try not to make too much fuss on marketing or anything around that. We just try to make it as easy and accessible as possible, but you can actually find all the events in the series. And as you see, if you were to see here, we had this Tuesday, we had an event in Paris, and today here, and next week we have one in Piracicaba in Sao Paulo in Brazil, and then one in Colorado, and one in Cali, and so on and so forth. And the last event of the year, as I mentioned, is in Queensland, Australia. And with that, thank you very much. Again, thank you, the organizers, for actually putting this event and willing to try it again once more. Thank you, the speakers, for taking your time to prepare the talks and to come and to share here in an event like this. And thank you all the participants, both here and online, for joining this. And hopefully, this will continue growing. So far there are limits. The limits are not inside us. So we are going to try to continue growing and to give this opportunity to more people in Europe, in Latin America, in Africa, in Asia. And hey, there's going to be one in Hawaii in the middle of the winter, so stay tuned if you want to take a break. Thank you.
So while they pull up the next presentation here, I'll introduce our first speaker. So sort of the theme is innovative technology and how it gets from the lab to the field and being widely accepted. So one of the goals was to have a speaker that could talk on an older technology, but that's still being used. And in this presentation will show us about mutation breeding and how it's still effective today and how it's still producing great results today. So Dr. Mitchell Tynstra, he is the Wickerham Chair of Excellence and the Scientific Director of Institute for Plant Science at Purdue University. And I got to know him when he was my undergrad advisor. So we're really glad that he took time out of his busy schedule to come over from Lafayette. So please welcome Dr. Tynstra. Just move the slides, PowerPoint, if you want. Either talking to that.
Right there. Sure. So I see.
So first off, thank you to the University of Illinois and for the graduate students that are organizing this symposium. It's a real honor for me to get an invitation, and I never say no to students. I always do whatever I can to come visit, because I think a lot of times as professors we get busy with thinking how important our research is, and I think one of the most important things that we do is we get a chance to work with students and train students that have an opportunity to go out and make a difference in the next generation of plant breeding and genetics. So thank you very much for inviting me. Thank you to DuPont Pioneer for sponsoring. I've had a chance to be involved in several of these events, and like I said, this is one of the nicer opportunities to get out and become involved with student organizations at different universities. So again, thank you very much. So I know Brian a little bit, and I guess you guys probably know Brian as well. So when Brian called, he says, we were wondering if you'd be willing to come speak at the symposium. He knows a little bit about my research and even a little bit about what I'll be talking about today. And he said, we were wondering if you'd be able to talk about mutation breeding. And this is I teach an introductory plant breeding course, grad plant breeding course right now in the fall semester. And just last week I was talking to the students about how unusual a time this is, where it's difficult for me to really anticipate what the future looks like 20 years from now. With the advent of gene editing, right now we're just barely getting into it. There's going to be fantastic breakthroughs in technology, both in the public and private sector, that are going to transform the way we knock genes in and knock genes out and recombine things. And then combine all of that with traditional plant breeding technology. It's a really exciting time to be someone who's thinking creatively about what can I do to create the next generation of plants that farmers can grow in farmers' fields. So that's really exciting. We're going to talk a little bit, I know, today about some of the gene editing technologies, about targeted reverse genetic strategies that can be used to manipulate genes in plants. And as Brian alluded to, this mutation breeding is really version 1.0 of gene knockouts. How do you create induced genetic variation that you might be able to use in a breeding program? And so that's what I'll be talking about today. And we're using it both from a gene discovery as well as a plant breeding perspective, and I'll talk about that. So the first thing I'm going to talk about is, just in terms of a general introduction, is something we call our functional gene discovery platform. This is a mutation-based gene discovery platform. I just want to give you a little bit of a description so that you understand what these programs are defined or derived from. And then I'm going to transition into what I use that platform in our trait development pipeline is what I call it. It's essentially a gene machine that allows you to target genes, or either in a forward or reverse genetics mode, manipulate them either for basic science or for plant breeding, and then move forward. And I'm going to give two examples of how we're using this research in my own program. We've got actually a lot of activities going on. I'm just going to provide an example where we're using a forward genetics approach to identify and deploy mutations that disrupt the secondary metabolism, metabolic pathway for durin production in sorghum. And then I'm going to talk about a reverse genetics approach to manipulate genes that are involved or that could be involved in grain quality attributes. And all of this is going to be done in sorghum. And sorghum is I think, a good example of a crop, the type of crop that's really going to benefit from gene editing and some of the new technologies, because until now it's not always been easy for some of the non-major crops to play in this space. And I can look at a crop like sorghum where there was a relatively modest investment in developing this pipeline and the impacts that it has in allowing myself and my colleagues, my scientists and students, to think of a clever idea, go sit down at lunchtime, do a little bit of computer work your way through a bit of some databases, and say, yeah, I think I should be able to do that in sorghum, and then come back out and tell your students, hey, here's an interesting idea, let's try to do that. And it's a really, the speed at which you can sort of think about ideas for moving forward is really exciting. So at Purdue, I work on C4 plants, so breeding C4 plants, translational genomics for C4 plants, and I work primarily on maize and sorghum. These are kissing cousins from the standpoint of crops. They look a lot alike. For the first 2 months, most people can't tell them apart before they flower. They have most of the same genes. A lot of them overlap between the two species. And one of the big advantages of having these two systems is that one can serve as the outgroup for the other. If you come up with a clever idea or an interesting genetic hypothesis in maize that may or may not be testable, or you want to come up with some additional data, I jump into sorghum and work on it on that side. And if I have something interesting in sorghum, I flip it back around and I look at it on maize and try to use those two systems to validate genetic hypotheses we might have. So what else? What are the differences? Well, maize originates in Mexico. Sorghum originates from Africa. Sorghum grows wherever it's too hot and dry to grow corn. Honestly, that's where it grows. As corn has become better adapted to drought conditions, it continues to move south and west into the drier parts of the western corn belt. Today, the line is about Manhattan, Kansas. North and east, you're in corn country. South and west, you're in sorghum country. And it's driven almost entirely by economic risk. How much input costs can the farmer justify in a given environment? Sorghum, if you look at this little panel of these panicles that I have shown here, this, I think, does a nice job of describing to some degree, or showing to some degree, the variability that exists in this crop. So sorghum exists as the domesticated sorghum crop, there's 5 major races that are geographically relatively distinct and represent a lot of the botanical variation that's available. So panicles 1, 3, and 4 are bicolor types. These are the least specialized of the races of sorghum. Oftentimes these aren't being used for grain production. They have other attributes that are valuable to farmers. In many cases they're used because they have sweet stems. They're grown in the corner of the field. The kids want their parents to grow a little bit of sweet sorghum so that if they don't have access to sweets or they don't have access to sugar cane, they can go out and snip stems. You know you're doing well as a visitor when the kid comes up, when you're at a small farm and a young person comes up and hands you a sorghum stalk, a little piece of sorghum stalk, because that's their version of the local sweet, and you're in when the children of the house are willing to share some of the things that are special to them. The second panicle here represents the Guinea race. It's from West Africa, uniquely adapted to the humid zone of West Africa, from Nigeria west all the way to Senegal. The panicle in the middle is a Kaffir from southern Africa. Kaffirs are really important from a production point of view because that race has contributed substantially to the development of females, seed parents that can be used in hybrid seed production. The next 2 are Caudatums. Those are important because they're from Central Africa, and they largely make up the male parents or the pollinators that are used for hybrid seed production. And over here on the far end, on the right, these are the Duras from East Africa. They all have unique botanical characteristics, and if I wanted to be a botanist, I could sit up here and talk about all of the botanical characteristics of the panicles and the seeds, but suffice it to say, there's a lot of variability. That's just in the panicle architecture, and in the plants themselves, there's tremendous variation in phenology and composition etc. Sorghum also has really a tremendous genomics resource, a really tremendous genomics toolbox. It's the most recent version of the genome shows it's got a little over 730 million base pairs compared to the 2.3 billion base pairs present in maize. It has about 34,000 genes that encode proteins, and it has 10 chromosomes. So in terms of the genetic resource, in terms of germplasm, it has a really tremendous crop in terms of genetic resource, genetic model. And again, I personally really enjoy offsetting these two crops, corn and sorghum, against each other because of the advantages that the different systems have in terms of doing research. So despite the fact that we know a lot about germplasm, I really love working in sorghum germplasm. Despite the fact that we have a reference genome, we know where the genes are, we think we know what the genes do compared with some other reference species in most cases, we don't really know that much about what those genes do in terms of how do they all contribute to the variation that we see and the variation maybe that we don't see and don't understand as well. And that's really the science of functional genomics. I'm not really a functional genomicist, but I play one in the lab sometimes when I'm trying to respond to hypotheses about what's driving the variability that I think is important. And it's important to me because I'm interested in functional traits that it can be used to improve the crop. In many cases, we don't have a very good understanding. Sorghum is almost, despite the fact that it's received a lot of investment recently, it's just barely better than an orphan crop. It just hasn't received a lot of research investment over time. So how do researchers try to connect genes and phenotypes? One common way that's used today is linkage and disequilibrium-based methodologies, whether it's GWAS or QTL analyses, different kinds of strategies that are used either in structured or unstructured populations to show an association between a trait and a region of the genome. So I'm sure many of you are aware, especially if you're a plant breeding or genetics student, here's a classic Manhattan plot, lots of pretty colored dots on a figure with the x-axis being the genome and the y-axis showing the significance of the association. In this case, I've plotted a little bit of QTL data along the top here. And so in a single figure, we can say, where do we see an association based on QTL analysis? Where do we see associations based on disequilibrium or association? And in this case, these are regions of the maize genome that are associated with variability in staging. And as awesome and as powerful as this is at sort of giving you insight into the genetic architecture of traits, complex traits, to me, this is mostly a hypothesis generator. Where are these regions? Can I dive into a region? Do I see candidate genes? Even if I see the best possible candidate, the most logical one that you could think of, you still can struggle to say, what's down here? What is driving this effect? This doesn't even make sense to me. And sometimes you have the opposite where it's like, this is exactly the opposite of what I would have expected based on published research. And so, as I said, it's a really powerful technique, and we largely use it for hypothesis generation. What does this tell us about architecture, and do we have candidates that are popping out of these analyses that might help us explain either a physiological relationship or a metabolic relationship, or depends a lot on what's driving these types of traits. The other strategy that geneticists have loved for decades has been mutants. So genetic mutants or variants are really powerful tools for demonstrating linkages, causality linkages between genes and phenotypes. The challenge is, do I have a mutant in the gene? So I can come up with lots of genes. Today, if you do a GWAS, it might spit out, depending on how stringent you are, might kick back 100 candidates, and it's like, man, 100 good ones, all of which look interesting. And you ask yourself, which of these is real? Which of these do I want to chase? Are any of these causal? Do any of these help me explain this phenomena that I care about, either I or my students, in the lab? And so some of what I'll be talking about today is our quandary, where now it's relatively easy to do linkage analyses or linkage-based gene discovery or hypothesis generation. We have an awesome genome. Now in sorghum, a lot of our discovery platform is around, can we routinely identify mutants and genes of interest that happen to pop out? And this can be done either using a forward genetics approach where I don't know what the gene is, but I'm interested in a phenotype and hammering our way through a mutant population, or flipping that around and say, I have a gene that's interesting for some reason, a gene sequence. I wonder what the phenotype is in reverse mode. And we use both of those strategies in different situations to try to inform either genetic studies or to create genetic resources that are useful in our breeding programs. So we took in sorghum, the reference genome is TX623. It's the best studied of the sorghum genotypes. So lots of new resources coming online, but it's the one that most people work on in terms of especially gene annotation and trying to understand physiology and biochemistry. So we treated the seeds with ethyl methyl sulfonate. We grew them out. We self-pollinated them for 4 generations to try to get as many of these mutations homozygous as possible. And we had a population of about 12,000 individuals. And when we grow those out, it's not difficult to find, if you don't have a plan and all you're doing is looking for variation, there's a lot of variation, and it can be variation in phenology and biochemistry and physiology and you name it. Every row is full of strange plants that have messed up genes. On average, we see a mutation in a trait of interest about every 800 genotypes. So we have, if you're looking for BMRs, there's 4 major genes. Brown midrib, for example, there's 4 major genes, and you'll find a brown midrib plant about every 200 rows. So the mutation density is relatively high. But we wanted to be able to use this resource in a more a more sophisticated way. We wanted to be able to, instead of not knowing what the mutations are, we went in and we randomly pulled 600 of these mutant lines and sequenced them at about the 6 to 8x depth so that we could identify the mutations. So we have a wonderful reference genome. We mutate the reference genome genotype, drive relatively inbred mutants out of that population, and then sequence 600 of them. And I worked with a couple of my colleagues at Purdue, Brian Dilks and Cliff Weil, to analyze this sequence data. And there's enough noise in the sequence data that you got to be careful about the signal-to-noise ratio, about how many things do you want to chase. And so, in our case, we created a pipeline, a SNP calling pipeline that had a few requirements. First, the SNPs needed to be homozygous. We can do some work with heterozygous SNPs, but it's a little noisier. Then we wanted the SNPs, if we wanted to have data for the SNPs, they had to be derived from data from both strands. Then we really narrowed it down and we only focused on the coding region. So we were only looking at the coding sequences. And lastly, since EMS causes G to A or C to T nucleotide mutations, we ignored everything except those. And when we do that, we would normally find about 100 missense and nonsense mutations in every line. So it turns out we had about 70,000 SNPs in genes that were either missense or nonsense mutations. And that then is our functional genomics platform. Sorghum has 35,000 genes, so we have a mutation or 2 for most of the genes that we might find as a candidate in whatever type of research. And so this is the platform. This data is available can be queried at this website that I show at the bottom here at Purdue, and we've deposited the collection at GRIN. And so if you find you can use the database to look for mutations in genes of interest, and then request the accession from GRIN. So it's publicly available. So don't let there be any doubt. I'm a plant breeder. I love genetics. I love studying genetics. I consider myself a geneticist, but I love plant breeding. And so my interest is in how do you use this resource, or at least one of my interests. How do you use a resource like this to do plant breeding? And that's what I came to talk about today. Mutation-based plant breeding. And so first I'm going to talk about a strategy where we're using a forward genetics approach to identify genes and alleles that eliminate cyanide production in sorghum. And then a little bit later, I'll talk about reverse genetics approaches to try to hit genes that might be involved in grain quality of sorghum. And then I'll do a wrap up. And so let's start with the forward genetics approach. So sorghum what did I say? Sorghum is grown. It's important. It's not grown. It's important to farmers everywhere it's too dry to grow corn. If a farmer can grow corn, you know what they grow? They grow corn. I don't know what it is about corn. Everybody loves corn everywhere in the world. The second the conditions or the environment changes to where there's an opportunity to grow corn, farmers grow corn. And it's normally at the expense of sorghum. So there's this interesting sort of conflict between these two crops. And you would think, because sorghum grows where it's hot and dry, grows under low input conditions, it grows in lots of terrible soils across Africa and South Asia, that it would be natural as a forage crop. So sorghum grows without very much water, doesn't require a lot of fertilizer, it's a low input crop, you don't have to have $200 or $300 of inputs to grow the crop, so it seems like it'd be natural for a forage crop, and it's important. There's about 15 million acres of forage sorghum grown globally. But one of the challenges of sorghum, not everybody grows sorghum, is that it produces a secondary metabolite called durin. So durin is a cyanogenic glucoside of sorghum that accumulates to incredibly high levels in young plants. Imagine a 3 to 4 leaf sorghum plant that's producing durin. It can actually accumulate levels of 10% dry weight durin. That single metabolite, 10% dry weight at the 3 to 4 leaf stage. And then from that point it doesn't accumulate so much, and it dilutes as the plant grows and ages. So young sorghum plants are incredibly toxic to herbivores. Because of this accumulation, they negatively impact forage quality, and every forage sorghum breeding program typically reports and markets their hybrids based on what's the potential, prussic acid, so hydrogen cyanide, also known as prussic acid. They're oftentimes marketed based on HCNP, hydrogen cyanide potential. And a lot of that's determined genetically, but if you do a lot of fertilizing or if you have adverse environmental conditions, they can also accumulate later in development high levels of hydrogen cyanide or durin that can be released as prussic acid. So how important is this? This is just little clippings out of the newspaper. It's important in Missouri. Dry weather can lead to a lethal lunch. Extension Service out of Arkansas. Heat and drought can create danger for cattle. AgriLife in Texas. Livestock producers should be aware of prussic acid levels in forages, particularly sorghum. Gainesville, Florida. Cattle deaths blamed on natural poisoning. Forage sorghum. Kansas City. Dry weather can lead to a lethal lunch. I guess it's another release of the same. Drought forces tough choices. Cattlemen feeling the heat as drought expands. Nebraska, grazing, Sudan grass, pearl millet, and sorghum hybrids. If you read the text down here, one rule that I suggest you never forget is to never feed hungry animals, or never turn hungry animals into Sudan grass or sorghum pastures, because they may eat rapidly and get a quick overdose of prussic acid and die. So this is very common. When I go across the Western Corn Belt and talk to cattlemen, people either dairy or beef, and say, have you ever had a problem? Do you know the frequency of yes, I've had a problem? Everyone has. So everybody knows about it, but they still grow 15 million acres of sorghum because it's a low input crop, it can be managed, it's usually ensiled, it's chopped and put into an ensiled to bleed off this cyanide gas. It's just that occasionally, every year, there are issues with cyanide, and you especially want to be careful if you want to use it as a hay crop. Or if you have it in your field and the cattle next door push through the fence at night and you come into the field the next morning and the cattle are dead because they've been in there all night and you weren't aware that this was going on. Interestingly, we know a lot about durin biochemistry. And so sorghum was actually the model crop that was used to describe this type of metabolism. Eric Kahn in the 70s wrote several beautiful papers. More recently, there continues to be a lot of work in sorghum looking at this trait, or at this metabolite. It's not really clear what it's doing there. Most people assume that it's there to deter insect herbivory in young plants, but it's not exactly clear. There's not really any good evidence. Some people think, oh, there's a lot of nitrogen in durin. Maybe it's the nitrogen, maybe it's the reason sorghum grows so well in low nitrogen soils. A lot of people have hypotheses about what's going on, but suffice it to say, for the moment, we know about the biochemistry, the metabolism. It starts with tyrosine, and it goes through those. It pushes through these two P450s, CYP79A1 and CYP71E1, and this glycosyl transferase to produce durin that has this cyanide moiety on it. It's not good for plants to be bleeding off cyanide into cells either. And so what they do in sorghum is they put this in the epidermis, and they put durinase, the enzyme that breaks it down, in the mesophyll cells. They put them in they accumulate in two separate cells, so they don't accidentally metabolize cyanide gas into the cell. The only time it's released is when a macerator, a chewer, an insect or a cow or something, chews up the leaves, the juices get mixed, and it's amazing if you have a plant that's 10% dry weight durin that's exposed to durinase, an extremely active enzyme, and it just blows cyanide gas to the point where it's very easily detectable. So I was interested in, I wonder if we can change durin metabolism in sorghum to produce a crop that's more favorable, better quality for animal producers. So we looked first at the natural variation, hundreds of landrace varieties, and we saw about a 5-fold variation represented in this panel, just a few of the accessions. TX623 is here. It's in a medium high. That's the reference genome of sorghum. We have some over here on the right that are quite a bit higher and some on the left that are quite a bit lower. Oops. And so we were wondering, I wonder what's causing the natural variation, this quantitative natural variation. We never found a null in the natural gene pool in the standing variation of sorghum. But we took these two varieties, SC1103, which was low, TX623, which was medium, sort of a high medium. We made a cross, and you look at them, they look quite different. This is a straight up Kaffir, a little bit of the caudatum. This is more of a Guinea bicolor, a lot of genetic diversity. We made a cross, we produced an F2, and then we plot that out in this histogram in the frequency distribution, and what we see is those plants that accumulate relatively high concentrations are bordered on one side by TX623 with a little bit of transgressive segregation, and on the left, approaching zero, but none of them zero, SC1103. So we have variation, relatively quantitative. I wonder what's driving this variation. So we know what the pathway is, so we go in and we look at the sequences of CYP79A1. It won't be quite as easy for me to suffice it to say, I'll give you a little funny aside because there's a funny story that I had. So I asked the student, there's actually 3 genes on the biosynthetic side. There's these two P450s, CYP79A1 and CYP71E1, and this glycosyl transferase. I said, go and look at the sequences of these genes in SC1103 and in the reference genome TX623 and see if we can find SNP variation that we can make markers for these genes. So then we can find out in the segregating population what's driving this variability. Do any of these drive this variability? And so the student went out, they looked at we generated the sequences. There's almost no coding sequence variation. Oh, we've got a little bit of variation in the promoter. Okay, we can create a SNP marker, a Caspar marker for the promoter regions of each of these genes. We go into the population, we extract the DNA. We go and use our markers for each of these genes. And he comes back to my office and says, they're all significantly linked. I'm like, what? That's a surprise. All of them? All 3 of them have a p-value of p is equal to 0.00001 level of significance on a single factor basis. And I'm like, that is very surprising. All 3 genes are linked to this phenotype. I said, are you sure? Yeah, I'm sure. I said, did you look to see where they are in the genome? Did you look just see where they're distributed? He said, no. Could you please go look? And he came back a half an hour later with a sheepish grin on his face and said, oh, they're all clustered together. They're on a 100 kb fragment of chromosome 1. So the markers were all segregating for the same fragment. And interestingly, these have come in as a single, almost like an operon practically, for durin production in sorghum. And the point being, with the quantitative variation that we see in SC1103 is controlled by it appears to be controlled by the biosynthetic pathway genes. I guess I was able to tell a story with no slides. I'm only going to talk about CYP79A1. So when we look at this SNP segregating this population, when we look at the SC1103, the low accumulator allele, they accumulate less than 1 micromole per gram fresh weight. And when we look at the homozygous alleles of TX623, they are high. If I remember correctly, I can see it actually, if I hurry up and look. 8.42 micromoles per gram fresh weight. So this is consistent with the biosynthetic pathway, for some reason, is driving the quantitative variation in Durin content that we see. But we didn't find any that were 0. That was really what I was interested in. So then we go and use our forward genetics approach in our mutant population. We grow these out in the sand benches in the greenhouse. I don't know how well you can see it. These are all little short rows of 10 to 20 plants. Some of them have albino mutants, oil yellow mutants, different kinds of things segregating. I actually used undergrads to do this. We would harvest the last ½ inch of the second leaf into a microtiter plate. We would freeze the titer plates overnight. The next morning we would expose them to Feigenlanger paper that we'd made. And Feigenlanger paper that's exposed to cyanide gas turns blue. And so what you see here, all of these little dots correspond to the wells of the microtiter plates. And so you can find which well has tissue in it producing cyanide gas, and occasionally you would see something like this and say, huh, that's weird. I wonder why there's no cyanide gas. There's lots of reasons why there might not be. So the students were always assigned, come up and talk to Dr. Tynstra, 4:45. I would always say, yeah, that looks interesting. Let's go to the greenhouse and take a look. And I'd say, which of the plants on the bench here was a 0? And they would point, and in some cases it would be a plant that was dead, almost dead, mutant, oil yellow, no way will ever survive. And I'd say, let's go home for the day. In other cases, they would point to a nice dark green vigorous plant, and I'd say, really? Are you sure? They, oh yeah, we're sure. And I'd say, okay, let's retry. So while we were standing there, I would retag the individual plant, take the next ½ inch off of that leaf, put it in a microtiter plate with controls, put it back in the freezer. I said, tomorrow morning at 8 o'clock, put it on Feigenlanger paper and show me the results first before you do anything else. And sometimes they'd come back and verify, wow, it's 0. And I'd be like, huh, amazing. Genetics is amazing, by the way. I say I'm not a geneticist, but that's always amazing. As simple as it can be, it's like, wow, it works. Genetics works. So we looked through 5,000 families, about 10 plants per family, 50,000 plants, looking for plants that couldn't produce cyanide gas. We found some that were mutated in the durinase. They could produce durin, but they couldn't release it as cyanide gas. Those were not super interesting because if you thought about, I want to improve the crop and feed the cows durin, but there's lots of stuff going on in the cow's gut, they could probably still release cyanide gas if you weren't careful. So those weren't very interesting, but what was interesting is we found 2 mutants, family 2447 and 5085, that were unable to release cyanide gas because they weren't able to produce durin. And so within this first family, when we look at segregating population, the F2 plants that are homozygous are 24 micromoles, and the ones that are minus are 0. The other family, 30 versus 0, and in the wild-type check, about the same. So then we extracted the DNA, we sequenced these genes, and found out they had the exact same SNP. Amino acid 493 is a C to Y mutation. I could probably spend more time talking about why they're exactly the same, but you can ask me at coffee time or something why they're the same, most likely why they're the same. So we had the same mutation, and it completely disrupts durin production. And the question became, is it possible to produce durin-free sorghum? Is that a real? We looked through hundreds of landrace varieties and didn't find any that were knocked out. Why were they not knocked out? Maybe it's important. Maybe it's key. Maybe it's key to the survival of sorghum. Who knows? One group in Australia had done some work with tilling identified a mutant that was knocked out, and the plant was kind of sick. And they stopped working on that particular mutant, and that was a little bit of it took some hope to push through that data. And there's a Professor John Burke at the Stress Lab in Texas who works on stay green, late season drought tolerance of sorghum, key adaptation trait to sorghum. And it turns out that Durin is a biomarker for stay green. So the first conference that I was at talking about this, he immediately corralled me and says, "Are you crazy? Don't knock out Durin, because that's key to, the trait that makes sorghum what it is, drought tolerant, is don't knock that one out or all you have is corn." Nobody laughs. Anyway, so we used that data and asked a few questions. We made a cross between 2447 and TX623 to produce an F2. We wanted to segregate out the background effects the best we could. And when we compared the wild type segregates versus the mutant segregates for the expression of green leaf using a chlorophyll meter, a CCM 200, at 3, 6, and 12, 3, 6, 12, and 18 weeks, there wasn't very much variation. And if we did the same kind of experiment with a cross to another line, they're all about the same. There wasn't a difference in greenness in the fields using at different stages of plant development. Well, did they grow at different rates? We did the exact same populations. We harvested the seedlings at different stages of development. And there was a little bit of variation, but one might attribute it to background effects or maybe not real. Who knows? But at least it wasn't a big, substantial impact on the growth of the plant. So we used this data then to say, look, we're going to have to act, if we really want to test the hypothesis of can we produce cyanide-free sorghum, let's get them in a breeding program and start working through this. And so one of the reasons I really like this project, it's my own hobby project. It's the one they let the professor work on over on the side of the field. That's where I'm usually working. And the person on the biomass harvester is Andy Linville. So we began working on developing hybrid grain sorghum, forage sorghum, and sorghum Sudan that had the cyanide-free mutations. And in 2016, we had our first hybrid. In 2017, we actually have a large number of hybrids. And so what we have is we've got these different classes: grain sorghum, sorghum Sudans. These are a hay type of sorghum. Forage sorghum that are optimized for biomass production. And we don't have a photoperiod sensitive type yet, but we're moving that way. We should have one in the next year or two. All of the ones are red, so red means stop. Those are the commercial checks that produce cyanide. So Alta and Richardson and Sorghum Partners have commercial hybrid checks in this experiment. They're all of the ones that produce cyanide, so yes, 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 yes. Everything that we're trying to do is in BMR, so brown midrib, modified lignin, highly digestible, good for cattle. And then we look at dry weight at 70 days after flowering and at 100 days after flowering. And what we see is we don't have anything good to compare the grains to, but when we look at the sorghum Sudans, they're all very favorable. They're better than the commercial checks. When we look at the forages, our cyanide-free forage hybrids are in the middle of the pipe. And we don't have our photoperiod sensitive sorghums yet, but we'll have those in the next couple of years. And so getting to the point of, can these be competitive for biomass production, the data says yes. And so we're quite excited that this is a new, we now are growing them across the US and variety trials across the states, and we still are looking for when is that thing going to happen, that unexpected, these haven't been in the, as far as we can tell, haven't been around before. When are we going to run into that situation where, why is cyanide there? Oh, that's why, because it's doing something really important. We haven't found what that something important is yet. It probably does something important, but we haven't quite found what it is. But this is an example of a forward genetics approach in mutation breeding. How about reverse genetics approach? And it works in forward genetics mode. This is very important because our assay costs 0.4 cents per phenotype. It's cheap, it's fast, I can hire undergrads to do it. They'll work for pizza practically, and I even pay them by the hour as well. But a lot of other traits aren't that easy. And that's where reverse genetics is particularly valuable, is when you have a not easy trait. So we went into our collection of 600 sequenced mutants, and we found 65 lines that had mutations in starch biosynthetic genes. This is just a short example, and this is what gets spit out of this database. These are the EMS numbers. These are the sorbic genes. These are the mutations. Some of them are missense, some of them are nonsense. For example, W521 stop codon. The asterisk is a stop codon. So a lot of these are large effect mutations. We're in sorghum, so I wanted to know what the maize annotation was. What do the maize guys think this is? And then where is it at in the genome? So this is just a small subset, and what are they? There's starch synthases and starch branching enzymes and starch debranching enzymes. All of the starch manipulators that we know about. Not in sorghum, but in potato and in rice and in barley, crops that have more money to invest in grain quality, because there is, for the most part, no money invested in grain quality in sorghum. So how would we use this kind of information? One of the points that we're interested in is, can we start creating value-added specialty traits for sorghum grain? So sorghum is unusual in its starch in that it doesn't gelatinize until almost 75 degrees. That's almost 10 degrees higher than the other cereals, and that's a problem from a processing point of view. I'm not a food scientist either, so don't ask me any deep questions about starch biochemistry. But it's a problem. A lot of sorghum is used for making beer. And if I have to push the temperature all the way up to 76 degrees, I'm going to do a lot of bad things to the alpha amylases and the amylases there, because I will have killed them and denatured them during the gelatinization process. Barley is more like 63, 64, 65. And so having high gelatinization temperature is a bit of a problem, and we're wondering, is this a target that we can manipulate in sorghum? Unfortunately, we know almost nothing about this in sorghum. Practically nothing. But we do know something about this in rice. A lot of work has been done on rice quality. People care a lot about rice quality. Not everybody agrees on what quality rice is, but they care about quality for whatever my expectation is. And it's known that ALK1 in rice influences starch gelatinization temperature. And so the question is, do we have ALK1 in sorghum? Indeed we do. Do we have any mutations in ALK1 in sorghum? We do. SBEMS 4308, this is its SOBIC number, this is its mutation, it's a starch synthase 2A on chromosome 10. It takes a lot of time to measure starch gelatinization, to process, to extract the starch, and do a lot of the work on starch. This is a good example of, I don't want to look at 50,000 plants, I'd prefer to look at a small handful. So we extracted starch from the reference, TX623, as well as the mutant. And I wanted to highlight this. Look at what happened to the starch gelatinization. Peak starch gelatinization temperature. No selection by us, only using reverse genetics to find mutants that are disrupted in a gene in rice that's known to influence starch gelatinization temperature. We look at a couple of lines that have funny genes in starch biochemistry or starch metabolism, and with almost a small handful of samples, 10 samples in that first experiment, we find one that's shifted by 13 degrees Celsius. That's the power of reverse genetics, especially in a species that doesn't have hardly any data on grain quality. And so again, you think about potato and rice and corn and barley, crops that are important for the commercial food sector. A lot of work has been done. And we're using all of the candidate genes that have ever been identified in these other species now to go into our functional genomics platform. Do we have a mutation? We don't always have mutations, but we do a lot of the time. Pull those back, talk to our food scientists and say, we don't need to look at thousands, but can you help us look at these 10 samples? And all kinds of interesting phenotypes start popping out that look just like they look in rice and barley, and maybe not so much in potato because sorghum grows on the head, but in many other species. They're very interesting. So we're extending this work now by developing a library of mutations in starch synthase 2A. We have now 11 different mutations that influence this trait in 3 different genes. And now we can start playing this game of looking at single gene and multi-gene mutant stacks to find out what happens to starch and starch quality in this, practically this orphan crop, relying heavily on the genetic information of other species. And from a plant breeding point of view, this line that's over here on the right, it's called Massia. It's used widely for brewing beer, for malting and brewing beer in Africa. It's also important in the food-grade sorghum industry in the United States, an expanding industry. And so this is where we do our proof of concept. And at the same time, we're incorporating these traits into African land races that are important in different regions where we do our work as well. Just a few closing thoughts. There's a lot of opportunity a lot of opportunity to do plant breeding, to do public good, to enhance the crops that we work with today, crops that historically have been almost forgotten, by using the research investments of other crops to improve the product quality, grain quality, sustainability of plants and crops that are important in different parts of the world. And we're going to have to continue working on our major crops as well as in our orphan crops or less important crops. We'll just call them less important crops, at least to us maybe. They're important to somebody. And so there's lots of opportunity, and a lot of these new tools and gene editing and other types of biotechnologies are very, very promising for looking at future adaptations to these species. A couple of quick words. Cliff Weil is there on the top left and Brian Dilks on the bottom left. We're really key collaborators in developing the sorghum mutant population and functional genomics resource. Andy Linville on the top right there is he's the person who actually does almost all the work in the breeding program. He was talking to me this morning, Mitch, what about this? What about this? When are you coming back? We got to get this work done. It's time to harvest. When are you done with the students in Illinois? I said, I'll be back on Monday. Kartik Krathapalli was important in establishing the screen for cyanide production in sorghum. And Molly McKnight on the bottom right did a lot of the early work in grain quality in doing reverse genetics for starch gelatinization. She now works at KSU. So with that, thank you very much for your attention. There it is.
All right, well, it's important to hear. It's important to me. Yeah, we have questions now. Let's see what in the back.
So my question is that, in the plant there are always multiple genes that can produce the same thing. How come that only one change that could cause this entire thing? Because for a lot of important metabolites, there are multiple genes.
In this particular case, there's not genetic redundancy. Sorghum doesn't have the genetic redundancy of maize. And so a lot of times when we try to do functional genomics in maize by transposon tagging or whatever strategy we want to use, more often than not we're disappointed by, boy, that looked like a promising candidate, but when we knock it out, it doesn't have a very obvious phenotype. In sorghum, it's not a derivative of recent ploidization. And so most of the genes in sorghum are single copy genes. And so that is actually one of the reasons why we play with corn and sorghum as partners in terms of genetic partners, is because it helps us to test some of these, especially the functional genomics hypotheses in many cases more precisely.
If I understand correctly, that during is a fairly substantial nitrogen sink. When you eliminate the production, do you have any idea where that extra nitrogen goes?
We're actually working with biochemists, because you can imagine what happens to the biochemistry of the plant where you have optimization of those pools of substrates. We're interested in that question, and we don't have very good answers, but I'm sure it goes berserk when 10% of the dry weight all of a sudden gets freed up back into the plant. But we don't see any funny, they don't grow faster, they don't grow slower, they don't turn yellow, they don't turn white, they're not darker green, they're not less green, we don't know.
I would expect you to increase nitrogen pool, you'd have higher photosynthetic capacity, more yield. I mean, yield is spot on. Just like wildlife. So I'm really curious where that nitrogen went to.
I don't have a good answer to that question yet. We're doing some looking metabolically, actually.
So you make it sound like you're almost waiting for the foot to fall in terms of some of these endurance-free maintenance. I mean, do you have any inkling at all that like increased pesticide requirements or anything at all that you've seen that would suggest that evolved during in terms of the viability of the plant?
So one possibility, Durin accumulates, its maximum concentration is at the 3 to 4 leaf stage, and by the time they're 1 meter, you can turn cows out on them. So it's not very toxic at the 1 meter plus stage. Almost all of the sorghum grown in a sort of a commercial environment in the United States is going to be treated with Cruiser or Poncho or any one of these other seed treatments that's effective for the first 60 days. We haven't been doing that. We've been leaving them exposed without insecticide treatment because I'm looking for it. And even looking for it, I don't see it. I thought, well, let's get out of Indiana into Texas where there's a lot more sorghum grown and we'll see a lot more pressure, but we haven't seen it. And if we do see it, my response is going to be, if it occurs at the early stages, we have a seed treatment. I mean, it's almost automatic. Farmers won't plant sorghum without seed treatment, and so that would be the likely way to deal with it in a commercial environment.
Awesome. Well, again, join me in thanking Dr. Townsend. Two reminders. We will have a panel discussion as our keynote session. So if you have further questions, feel free to attend that or post your questions online for that. As well, there's a mixer at Crane Alley at the end of after the symposium at 6. So if you have other questions, hopefully our speakers will be there to quiz over a beer.
Okay. Good morning, everyone. It's my pleasure to introduce you to the next speaker, Dr. Maria Fedorova. Dr. Fedorova received her PhD at the Institute of Agriculture Microbiology in St. Petersburg, Russia, and in the United States, she's been as a postdoc at the University of Minnesota before she joined DuPont Pioneer. And now she is the regulatory strategy lead for DuPont Pioneer, and she's gonna introduce us her presentation about CRISPR-Cas. And please join me to welcome Dr. Fedorova.
Thank you very much for the organizers. Can you hear me? Okay, good. Thank you very much for the organizers. I was really excited to see the topic of this symposium coming out of academia. It is not only about cool scientific innovations, it's also looks like you really appreciate other factors that could influence the great idea that you have in your lab, whether it will see the light of the day, whether it will make any implication to the society. From the topic of the symposium, you realize that public acceptance, public perspective, or policies governing your technologies or your products can make a huge difference whether this cool idea will see the light of the day or not. So my presentation is not going to be heavily technical. I can take some technical questions. If I don't know the answer, I'd be happy to follow up with my colleagues at DuPont Pioneer on the technical experts of CRISPR-Cas, but I really want to impress you upon the opportunities from the technical perspective, but also to talk about what other things do matter to make a difference with this technology. And I have my plan B, should the light go off here. So I hope I will be able to explain with hands what I want to say. I don't know your level of familiarity with the CRISPR-Cas gene editing. I talk a lot with various audiences and it's different, so I'm planning to do really high-level overview history, although it's even strange to say history because the technology is just pretty much 5 years old in terms of technology, not the biological application. So although if I said that it is 5-year-old, but in fact the knowledge about CRISPR has been about 20-year-old, where since late 80s, early 90s, once whole genome sequences become available and bacterial genomes were sequenced, scientists noticed very strange repetitive genetic DNA sequences within bacterial genomes, but they could not immediately explain what's the function of those sequences. So that has been held as a sort of mystery till much later, 20 years later. Danish researchers, they were able to understand what those repetitive CRISPR repeats mean in the bacterial genomes, and they figure out that basically it represents a sort of immune system of bacteria, the memory of the previous phage infection. So they are ready to fight those infections if it recurs back with the bacterial culture. And as you can understand, that's a very important practical questions, you know, considering how much bacteria is being used, for example, for yogurt production or cheese production and other things. So that was a really great practical breakthrough to be able to explain what those CRISPR elements do in the organism. But the real breakthrough in terms of CRISPR-Cas gene editing or genome editing as a technology didn't come till two big works that were published a few months apart from each other in 2012. And basically these works were about how we can harness that biological system, the biological defense system in bacteria, and to make it being used as a tool for doing similar things, which is cutting DNA, introducing DNA double-strand breaks in other systems, whether it is plants, animals, or humans. So the CRISPR-Cas as a technology, it started with 2012. And as you can see from this survey of scientific publications on CRISPR-Cas, since 2012, there has been big increase in the number of scientific publications on different aspects of CRISPR-Cas. And certainly this does not represent just plant scientists. That's an overall publication that includes a lot of interest in the medical field for treating diseases. This continues to rise because you see the 2017 numbers, they are just up through September. I'm pretty sure by the end of the year, it will be as equal as 2016 publications or probably even higher. Publications come out every day pretty much. So what CRISPR-Cas is, and in fact, when we talk about CRISPR-Cas for gene editing as a tool, it's a little misleading title because there's no CRISPRs in CRISPR-Cas as a gene editing tool. It's basically based on the Cas or Cas9 enzyme, which is most popularly known, although there are other Cas enzymes have been discovered since then. But essentially what CRISPR-Cas as gene editing system does, it is sort of a molecular scissors that only purpose is to introduce a double-strand break in the DNA sequence. So essentially it's a nuclease, it's a DNA cutting enzyme, you know, very similar in a way to what all famous or familiar enzymes do, ECOR1, HindA3, etc. So the difference from these enzymes is that it is also a targeted sequence-specific DNA break, but the sequence specificity is determined by the RNA or guide RNA that is sort of attached to the Cas9 enzymes, and it recognizes the motif from 17 to 20 base pairs. So we're talking about much larger recognition site compared to what we commonly use as restriction enzymes. So this specificity can be programmed with the use of the guide RNA so that it brings Cas9 nuclease to that point and cuts DNA. So that's essentially the function or the purpose of the CRISPR-Cas as a cutting tool. Now, definitely the DNA can stay unbroken there, so it has to be repaired. And based on what type of repair follows this DNA double-strand break, it determines several CRISPR-Cas applications of the system. Again, apologies if you know all of this, but I wanted to repeat for the sake for the whole audience. So there are three major types of applications based on the DNA break repair. They're often referred to gene off, gene edit, or gene insert. The other synonyms you could see in the literature as SDN1, SDN2, and SDN3. And SDN stands for site-directed nuclease, which is essentially CRISPR-Cas is. But I think it's important to keep in mind that that's not a unique gene editing tool. I mean, it comes after zinc finger nucleases, after talents, meganucleases. There are other things that were developed after CRISPR-Cas. They're very similar to that. CRISPR-CPF1, that's one of the newer enzymes that essentially fulfill the similar function as Cas9. So, in the simplest situation of the repair of the DNA double-strand break, essentially let it repair itself. There are a variety of plant endogenous native cellular mechanisms that exist in the plant to repair DNA. One of them is called non-homologous end joining, where DNA kind of stitched back itself. Sometimes it's a perfect repair, so you don't see any changes around the repair site. There's no additional deletions or additions of the nucleotides, which freely, available from the free pool there. But sometimes this mechanism is error prone. So sometimes that repair back with this non-homologous end joining results in a loss or an addition of a base pair. If it happens to be in the protein coding sequence, if it happens to change frame or to change amino acid and basically makes that protein non-functional, what you end up is with a gene off or gene knockout application. So you can knock out the gene. The other two applications of CRISPR-Cas is where you could provide a template how to repair that DNA break. In case of the gene edit application, the repair template is usually small, and for example, you want to target 1 or 2 specific amino acid changes. I'll give you some examples later on in my presentation. So in that case, that template DNA, which is homologous to your target DNA with the exception of this targeted change, targeted edit, would be incorporated into the plant, and you would have an edited version. And as I said, I'll give you some examples later on. So this is what is called a gene edit application. And in the third type of CRISPR-Cas application is you also provide a repair template, but for instance, you want to insert an enhancer element into the genome, or in more sort of extreme case, you want to insert a different gene into the plant. So that template would contain DNA sequence of that particular element or gene you want to insert, and the DNA break will be repaired through homologous recombination mechanism, but as a result, that would include the desired sequence into your genome. So I think there's a little bit of confusion in terminology, but with gene off, also called gene mutagenesis sometimes in scientific literature, gene edit, sometimes called gene correction, and insert, what we typically refer for gene editing is the gene off and gene edit application, because as you can see in both cases, this does not include insertion of any foreign genetic material or any creation of any new combination of genetic material that won't be possible through, for example, mutational breeding. Most of the current tools for generating gene-edited plants are based on when the components for gene editing, Cas9 template is needed, are delivered on plasmids, on DNA vectors, on plasmid vectors. And if we think about the process to generate gene-edited plants, it all starts with the guide RNA design. So essentially, you have to design these 17 to 20 nucleotide sequence to be specific to your target, where you want to make that targeted break. Transformation and regeneration follows that, and a key part in gene editing process is molecular analysis and characterization what you got. Definitely you want to confirm that you achieved the desired mutation or desired edit, but also very importantly, you want to confirm that if you used DNA vectors to deliver your gene editing tool, that none of that plasmid DNA helper tools are present in your plant before you advance further. So essentially, what you only have a plant with its targeted gene edit, and you have nothing else, no plasmid DNA from the tools in the genome. and only those lines would be advanced for future development. So essentially, what you end up with, no plasmid DNA. As I mentioned, those changes would be similarly possible through conventional mutation. So if you think about classical mutagenesis, EMS, irradiation mutagenesis, this is an untargeted mutagenesis tool. This is what we have with this CRISPR-Cas gene editing approach. It's a targeted mutagenesis. Of course, it's a knowledge-based. You have to start with the knowledge and information on the gene-phenotype relationship, what gene you want to target, what gene you want to change, and in what particular way, whether you want to knock it out, or whether you want to edit it a particular way to change and get the desired phenotype. There are also newer tools, I shouldn't say new, they're newer tools, but they're similar tools that have been recently developed. And in fact, you can deliver the gene-edited components right in the form of the protein or RNA, ribonucleoprotein complexes. So there is a potential even to avoid the DNA coding steps, the DNA delivery for gene editing. So core competencies that are needed for CRISPR-Cas deployment. First of all, obviously, CRISPR-Cas technology, and scientists very excited with the CRISPR-Cas. I mentioned that there is other early gene editing tools are still available. Zinc finger nucleases, talents, meganucleases, CRISPR-Cas. In Pioneer specifically, we've worked with all of them but zinc fingers, I believe. And our scientists are very excited with the flexibility and efficiency of CRISPR-Cas. So that's definitely a winner over. Oh, sorry. Thank you. Yes, I love to walk when I talk. The second important competency that one should have in practicing CRISPR-Cas technology is genomics. As I said, it's a knowledge-based tool. So you have to start with a knowledge of the sequence of your gene, what you want to modify, which way, and it's also you would want to have information on the whole genome sequence of your organism, because when you're designing and when you're, various software are available to design guide RNAs, so they will take account the whole genome sequence to find the most unique sequence in your target gene, so it would not introduce any off-target effect if this is of any concern. So bioinformatics is obviously a critical part of any sequencing or genomics pipeline, so this also needs to be available. And finally, targeting elite genetics. And I'll touch on that in the next few slides, but I think the reason why we call CRISPR-Cas as an advanced plant breeding technology is precisely because it allows us to make these changes directly into elite germplasms, not some model germplasms. As you know that, there's some recalcitrant crops for transformation, but even with corn and soybean, there is usually only a handful of varieties which are easy transformable, and the rest are hard cookies. And sure enough, if we think about elite germplasm, something that is more closer to the farmer's field, these are usually recalcitrant. So having an opportunity to introduce those gene edits as close to the elite material as possible is critical, because it saves time on trait development. One comment I want to make here is that sometimes I hear that CRISPR-Cas gene editing is portrayed as this is so fast, this is so quick technology, and it is true in terms of the trait development. I think it has to be recognized that trait evaluation process and breeding still takes several years. It's not like we make a gene edit today and we sell the product tomorrow. There is still several years of multi-locational evaluations of what we get to make sure we study things. It's a common breeding practice. It's no different, it's no special for gene editing. That has been done by plant breeders and commercial plant breeders for decades. So we're not making any shortcuts, cutoffs, expenses here. This is gene editing helps us facilitate trait development, get to the right trait very quickly, rather than different other types of breeding approaches. I'm going to touch on just really few examples of the gene editing or gene off and gene edit technique. And for the gene knockout technique, this is a future product, first product that Pioneer is currently developing. We hope to be on the market by the end of this decade. We call it CRISPR-Cas waxy corn, or next generation waxy corn. Sometimes you'll see it in our external materials. I don't know how many of you know about waxy corn in general, but I really want to briefly introduce the whole concept behind it. So waxy corn is a high amylopectin starch corn. Starch is composed from two polysaccharides, amylose and amylopectin. So if you look at the normal corn, or number 2 yellow dent corn, the commercial feed corn, it contains approximately 75% of amylopectin and about 25% of amylose. The kernel has this, very characteristic translucent appearance. This is the corn that is used for feed, ethanol production, as well as food. Waxy corn is not a new trait per se. Waxy corn has been known for over 100 years, and it is a kernel that looks like a candle. It has a candle wax appearance. The reason behind that is primarily all starch in that kernel is in the form of amylopectin. It's 95% of high percentage of amylopectin and just few percent of amylose. The reason being that waxy corn has a mutation in waxy1 gene. That's, again, waxy genes is one of the classical maize gene. They have been heavily studied. The gene encodes starch synthase enzyme, which essentially catalyzes production of amylose. And imagine if there's a mutation, any knockout mutation in the waxy1 gene, it results in no amylose production by waxy1, and as a result, a high amylopectin starch content. There is over 200 mutants, I think, that are known in various mutations in the waxy gene that results in that knockout phenotype. So again, waxy corn, it's not a new concept, and it's actually has been a commercial product for Pioneer as well as other companies since mid-90s, 80s. In fact, going back to the history, I think the whole interest to waxy corn here in the United States came during Second World War, when imports of tapioca and other kind of forms of starch from tropics were kind of challenged due to the wartime. So there was a reason to look for sort of a local solution for starch. And that's how waxy corn, which has been known even back before that, got its commercial future and started to be cultivated in the United States since basically Second World War. Okay, I want to make sure. So Pioneer actually is the leader on the waxy corn market. It's a specialty corn, as you'd imagine, because it has these particular properties of the waxy corn starch. And if you have this waxy corn starch, it's really valuable commodity due to its physicochemical properties, because it has, it better behaves with a freeze-thaw situation, so it's heavily used in, frozen foods. It helps with creaminess. It's used in sauces, salad dressing, etc., etc. So it has a huge food use. It also has some industrial use for paper production. If you get your Sunday newspaper full of, all these advertisements separate, and it's kind of glossy paper, most likely waxy cornstarch was used to produce this glossy newspaper, ads in the newspaper. So anyways, Pioneer has been, as I said, has been selling waxy corn, which is based on a particular mutation in the waxy gene. And when sequencing technologies became available, I realized that this mutation is due to the gene missing 30 base pairs in the middle of the gene, exactly intronic zone border. So don't divide by 3 and say like, oh, you just missed 10 amino acid, not exactly. We have been selling this since mid-80s. It doesn't come without challenges. So if we think about waxy corn, where it goes to the wet millers who produce waxy cornstarch. they don't want to mix waxy cornstarch with a normal starch because it will kind of, you don't have any value from that anymore. So they really want to have a stable supply of waxy grain every year from the growers to have to deliver that waxy cornstarch. Well, because it's an old commercial variety, we have to introduce, when we come with new hybrids, we have to move this mutation, and the mutation is recessive, so you have to make it on both sides of the pedigree. You have to make it available in both male and female inbreds. We have to do it by conventional breeding. We have to do 5 cycles of breeding to make sure we got rid of as much as genetic linkage drag as possible, so we don't carry inferior parts from the previous genotype into high-yielding varieties. And that becomes like a catch-up game, definite catch-up game, because by the time we finish introgressing this waxy mutation, it what was an elite inbred, guess what, it's not an elite inbred anymore. And the farmer, they're not scientists, they are not comparing to the right comparator. They say, this is great, but we like your current hybrid, which is P1197, whatever. Can you make this waxy mutation in your most highly yielding hybrid today? We say yes, and go back and do this do this introgression via conventional breeding, and the story repeats. So there is always a time lag to deliver that into the best yielding germplasms. And sometimes we can't fully eliminate some yield penalty that would be associated with this remainder of material through the breeding. So there's sometimes a linkage drag, and we attribute there may be some yield challenges that we see once in a while. They are due to this factor. So the solution is, if we can use CRISPR-Cas technology to introduce that waxy mutation directly into elite inbred, we can really save time and have much better yielding germplasm delivered to the farmer, as well as we hope, our linkage, our yield penalties once in a while that were related to the breeding program, so we can hopefully eliminate that as well. So that was the concept behind how we want to improve our current product using CRISPR-Cas technology. So what we used to do, we could perfectly replicate the 30 base pair deletion in the middle of the gene. What we chose to do is we actually chose to delete the entire gene. So we had to use 2 guide RNAs in that process. One cut in the promoter region of the waxy gene, upstream of the transcription start site, and the other cut in the terminator region of the gene. So using 2 guide RNAs, we kind of chop out the entire gene. Regardless of the particular mutation, all the downstream things are exactly the same. Gene is knocked out, well, not present in the second case, which results in the starch composition with pretty much everything amylopectin and a waxy corn phenotype. The gene edit application example, I wanted to give you 2 examples of the gene edit application of the technology. The first is really what I talked about when we're using a template DNA that contains just one little change. We want to do one specific edit in the plant gene to change its function. And I think the title of that, or subtitle of that slide, Reproducing ALS Mutant Allele Using CRISPR-Cas Gene Editing, it's precisely what we did, because this is an example where we used essentially a model gene, acetylactate synthase, model because it's the gene and protein, in fact, which is known in very many species. And there are also mutations in this gene that are available in very many species, and we know that particular mutation result in tolerance to an SU class of herbicides. So we wanted to use CRISPR-Cas to see, can we replicate that exact same mutation that is available and make it as specific as needed, because there is one particular mutation or one particular change in I don't have the number of amino acids, doesn't matter. Change C to T will change will make a change from proline to serine, and that will result in the herbicide tolerance of the plant. So what was a complicated factor in that case is that, in fact, plant maize in that particular case has 2 ALS genes. They are located on different chromosomes, and they're very, very similar to each other. I can't remember the percent of homology, but they're very similar. So the challenge was that we wanted to make a change in ALS2 gene, but do not touch ALS1, because it's still a very important gene for the plant, so we didn't want to knock out both genes in the plant. Not getting into the technical detail how that was accomplished, you can refer to this paper if you're interested, but that was possible to change. So we can selectively target ALS2 and don't touch ALS1 with this specific amino acid change, and you can see that it results in the gene type as expected. I mentioned that already, how we can use gene editing to facilitate breeding, and as I understand, most of you have breeding background, so I don't need to repeat that over and over, rather than to reiterate that instead of conventional breeding by backcrossing, when you try to introduce your gene of interest into your germplasm of interest and bring as less of other accompanying genetic material with you, it requires several backcrossing. It takes time. With CRISPR-Cas, we can move the desired allele of the gene and replace non-desired allele in our germplasm interest in pretty much one cycle, followed by several cycles of finishing and evaluation. So this is why we call that CRISPR-Cas enables us to accelerate breeding processes. I have only one slide just to give you examples of traits developed by gene editing. As I mentioned, the breakthrough to explain how we can use gene editing in general to edit as a tool came about 5 years ago. But no week passes by when I don't see new publication on CRISPR-Cas, and I'm not touching the medical application. I'm just looking at the application of CRISPR-Cas in agriculture. And that slide kind of aimed to hopefully impress you upon the opportunities, and that could take the entire presentation and so on. But maybe some key points I want to mention around here, and I'll be happy to provide any literature references if you are interested in any particular research here. But these are all traits that have commercial applicability. You can see that this slide lists both row crops as well as fruits, vegetables, trees. I think it's important also to mention that here with gene editing, the idea is to tap into plants' own potential. So you have to think in what trait areas plants really do have the diversity and genetic potential that we can capitalize on. And one thing that comes first probably is resistant to diseases or viruses. So you can think how many options and genes and pathways plants have that control various aspects of plant pathogen interaction or plant viral interaction, and you can see how we can use this knowledge to make gene editing to bring this trait much easier to you. Another thing I want to mention around that, and I think Dr. Tenster touched on that in his presentation, I think this tool is extremely valuable for crops with limited genetic diversity or for polyploid crops, because classical breeding tools for polyploid crops or crops with limited genetic diversity are really extremely limited. Most of the mutations have a recessive nature in the first place, right? So you have to find a mutation that happens in all 4, 3, whatever number of genomes it has. Using gene editing tools, it's relatively easy to introduce desired mutations into polyploid genomes, so you have it in all genomes, so it's functional. So this is a separate not a separate application, but I think it holds an extremely good potential for these type of crops as well. So again, I have to update this slide every week or 2 weeks when I talk, because I see great new applications, potential applications of the technology. And again, it's row crops, fruits and vegetables, industrial crops. If you look at camelina, for example, which is a source of industrial oil, or I think sitare is also kind of a bioenergy crop. So this is to be continued, and I'm really happy to see that. And again, I talked several times there about that, is like what makes gene editing, SDN1 and SDN2 applications, really similar to mutagenic applications. And as we know that mutagenesis and genetic diversity, they are fundamental for evolutions and crop breeding. And as we think about products of classical mutational breeding, they have tremendous history of safe use. There is a database of mutant varieties that's available, and if you look at it, it's over 3,000 entries of cultivars have been used commercially, the mutant cultivars that have been used commercially. So great advantage of gene editing is that it's a really targeted mutagenesis application, because for the first time it allows breeders to target mutation to a particular gene, particular genetic sequence. And that's why we really like to emphasize that this is a really yet another tool in the breeder's toolbox to get to the desired trait. And this is what, do I still have time? I hope so. Yes. So this is what I really want to touch upon, innovation. So let's step for a second from all the technical advancements and all excellent technical opportunities that this technology allows us to do. Whether it is science, whether it's technical capabilities of a given laboratory or a breeding company to do CRISPR-Cas, intellectual property, that's always, a question and a discussion topic. Collaborations, that's important. But what else enables innovation? This is one of these word cloud I found on internet, and I was looking for those word clouds for innovation, and there were not many where I saw things beyond science. You can see here innovation is revolution, leadership, research, intellect, development. I don't see those word clouds often. They will say policy, public acceptance, or social license, how we call it. But I think these are critically important components of any innovation, not just CRISPR-Cas gene editing, which would enable to realize all the brilliant ideas that are in the lab. With the policy, what I mean by whether countries, and I'm not talking about just US, but whether countries are ready to accept this innovation and to have an appropriate regulatory oversight, first of all, to determine whether it's needed, and if it's needed, to what extent it is needed. So on one hand, it will keep protecting public health, it will keep protecting environment, but it will not kill, it will not stifle the innovation. So what's the appropriate regulatory framework for any innovative technology or product? And as I said, social license. What are the social value? Do you trust? Who benefits the technology? Is the technology developer or technology user transparent enough with the society? So these are very, very important topics that will enable any brilliant idea that you may have. And I'll touch first about and talk a little bit about regulatory policies. And I think one of the most important parts, I think, on this slide is actually showing the globe. Because what I feel sometimes come very often here with the audience is that if we talk about innovation that is sort of US made, especially if it refers to a trait for a farmer, it's not only the US regulatory policy that matters. When yields, when grain is harvested from fields in Iowa, Illinois, any place else in the United States, it can end up anywhere. There is a global trade. It can go to China, it can go to Europe, it can go to Japan. So we need to make sure that regulatory policies are appropriate and adequate, not only in the United States, but they're harmonized and coordinated all over the world so we can really enable the trade. Well, for gene editing specifically, regulatory policies are yet in development in many countries. They are not have been established there, and it's a very simple reason why. So as you know, GMO crops, transgenic crops, are regulated in most countries of the world. And most of the countries chose to regulate those crops based on the process. So when the genetic engineering technology came about in 1980s, it was new. It was kind of perceived a little bit dangerous or unknown for the most parts. So most of the countries adopted a Cartagena protocol for biosafety, which basically says that GMO is when you use the recombinant DNA technology, and what you have at the end is the plant with a novel combination of genetic material. If you think what first GMOs were, they were insect protection or herbicide tolerance when a bacterial gene was inserted into the plant. Well, I guess you cannot obtain it through sexual crossing, so it is a novel combination of genetic material. It is when a recombinant DNA technology is used. So I think, up until now, it was a pretty clear case what constitutes a GMO and what needs to be regulated due to all these, because of all these legal frameworks in all the countries. So how about gene editing? I explained, I just showed the examples to you that we may use recombinant DNA technology at the front end of the process, but what we end up is the plant that doesn't contain any foreign DNA, and it can be a similar mutation that you've seen from conventionally bred plant or mutagenized plant, or from the plant that you found in nature, the inherent genetic variability. So that's why it sort of presents sort of a gray zone. How would you interpret the status of gene-edited product given the current GMO legislations all over the world? And the discussions right now, they're very actively happening among the governments, because everybody realizes that gene editing technology and products are right around the corner. How we would treat gene-edited product? Would we consider it to be in scope of the GMO regulation, or because it's similarity to conventionally bred product, we would consider it to be similar to a conventionally bred varieties, and whatever regulations apply to conventionally bred varieties, the same obviously should apply for a gene-edited product. So these are the discussions that are happening globally, and we hope we can see some developments in the next few years. And on this slide, I really want to offer a few considerations on the regulatory policy. As a company, we support appropriate and science-based regulatory oversight. And if you think about gene-edited products that could be undistinguishable from plants produced through conventional breeding techniques, having a long history of safe use, in our opinion, they should be subjected to the same level of the regulatory oversight. Because again, it's not production method, it's the characteristics of the plant that determines its safety. So we feel that, again, regulation should be science-based, and that means that it needs to accommodate new scientific evidence and learning, because again, what GMO was back in 1980s, now there's so many different ways how we can use recombinant DNA technology. That's a new learning. Our understanding of natural genome variability, our understanding of, based from the sequencing tools, from the whole genome sequences that are obtained, that's immense. We have to account this new knowledge. Regulations need to be cost-effective. They need to be transparent. They need to be consistently applied and enforced, because no product developer, commercial developer, could work in the environment when regulations are not predictable and you don't know whether you would need to spend this amount of money or that amount of money on bringing product to the market. And as I mentioned, because we are one global community, regulations have to be globally harmonized. So we don't have a product, ideally, that is not regulated in a simple version in this country, but regulated in some place else. That's really detrimental to any business and any innovation. Public acceptance or social license I mentioned, that's a very important component. As with any technology, but I think for us, we are involved in plant scientists, and I think some of you are working maybe with GMOs or transgenic plants. You know that GMOs is a very sensitive topic, and there are supporters of GMOs, there are not so big supporters of GMOs. So we realize that any new technology requires social license. And we really, as a company, we really want to listen to both stakeholders, traditional, by traditional, I mean farmers, value chain organizations. Non-traditionals would be stakeholders like consumers and public. And admittedly, industry, I guess, has made a mistake early on with the advent of GMO products, because I think we were so focused on our immediate customers that were farmers that we didn't pay enough attention to the public, to the consumers, to the value chain. And these are very important voices, and we need to hear what their concerns, where do they see benefits of the technology, where do they see challenges or risks for them with this technology. So we need to incorporate all this knowledge and not to ignore certain stakeholders, but really be focused on only like-minded. And as a company, we had several conversations with those, non-traditional stakeholders. And I think when it is a respectful conversation, this is really helpful. This is not as hard as you might think if you come across somebody who is completely against GMOs, have very big reservations about gene editing. But I think having this opportunity to hear their concerns and to take them into account, this is very, very important. And this is not an easy discussion, evidently, but it's an ongoing discussion. And I mentioned who are the stakeholders. Again, it's a variety of individuals or groups of interest, starting with academia, public and private institutions, seed industry developers like Pioneer in particular, growers and commodity groups, grain trade and value chain, and consumers. If you think about consumers, what top questions around gene editing comes? Well, the first one is all about safety, right? Is gene-edited food safe? Is it healthy and nutritious? And this is understanding. And it's very interesting when you talk with consumers about safety, because if you talk about food safety, especially in developed countries, this is sort of, they start with the baseline zero risk. So I think in the developed countries, we are fed enough, we have enough food supply, we can make choices, this is respected, definitely, but I think we come with a zero risk. And I'm thinking about that in comparison with the medical application. And I mentioned that CRISPR-Cas gene editing has tremendous application in medicine, treating various forms of leukemia, for example. And I think this conversation would have been easy in the medical field, because sometimes CRISPR-Cas, it would be a situation of life and death for a patient. So I think those patients, I would assume, would be okay to take some risk, understanding that the benefit could be far greater than that. So our conversations again around food is really, challenged sometimes with this perception of zero risk. Environmentally safe and sustainable. I've seen, interestingly enough, recently I've seen a publication from Michigan State University who conducted the survey on various topics of food, and it was very surprising to me to see that I think only a third of the population really cared and wanted to pay more for the food if it would have been more environmentally safe and sustainable. That was a little disappointed for me from the US audience. Discussions around, is it another GMO? This is not a very discussion that goes very far, because I think it's sort of irrelevant discussion in a way. With all the new scientific knowledge, it really blurs the line what GMO is. What is GMO, what is a non-GMO? And I think you probably all heard about, very interesting publication from Jonathan Jones' labs a couple of years ago, who showed that there are thousands of genes in sweet potato that seems to have a bacterial origin. So is sweet potato a natural GMO? So I think it's a road to nowhere if we try to convince each other, is gene editing is a GMO or not? I think much more important questions that we need to ask ourselves, why this tool is needed. And I really want to emphasize the importance of why. I don't want my message to come across that, okay, CRISPR-Cas is just a wonderful tool, it's a new shiny toy for Pioneer or any other company who wants to drop everything else and just do like that. I think it's very important that CRISPR-Cas is one of the tools that helps with the productivity of the plant. We are not dropping breeding, we are not dropping development of biotech traits, crop protection solution, which is the most appropriate approach. to achieve productivity. Because if you ask the farmer, what's your number one concern, 3/4 of them will say it's a productivity. And by productivity, you can mean a variety of things: yield, resistant to diseases, resistant to insects, drought tolerance. So this is a really generic thing. So I think for us, it's very important to emphasize that we are working on CRISPR-Cas not because it's a new shiny toy, but because CRISPR-Cas could address some specific challenges in productivity. It's needed for solve some specific problems, whether they're global or some local, and this is why we use it. Finally, if I have a couple minutes, I can't track my time anymore, but I think I want to touch on the collaborative mindset, because innovation happens globally and agriculture only benefits from that. Not all researchers or those breeders who have some great ideas about bringing technology, bringing some trait, have, for example, capacity to do CRISPR-Cas in their lab. So I think the more we spread an opportunity for collaboration, the better it impacts the entire society. And as I mentioned, there are some global problems, there are also some local problems, like local pests, local diseases, that maybe not everyone would like to embark on, but this could be a really helpful tool. So I think working globally and deploying this technology across all crops, all traits, will really help with the innovation. It will help enable national, local developers in different countries to do this work and bring this solution. And this will help with regulatory policies, because I think if the country sees that there's a lot of opportunities for local academia or local breeding companies to deploy the technology, it would be much friendly regulatory policy, because unfortunately not always science-based. It has other things to consider. So with having this collaborative mindset, I think it's very, very important for innovation. I want to direct you to the Pioneer website, because we have thought very hard about open innovation in the CRISPR-Cas space in particular, and how we can leverage our own capabilities, whether it is with CRISPR-Cas tools or breeding germplasms with needs of others. So we have an open innovation concept where we invite collaborators on CRISPR-Cas technology in particular. And if you are interested, you can visit that website and look for details. Who are potential collaborators? You can see, and you can probably find yourself here, but it's a really broad set of folks we feel we could work together to enable with this technology, and the terms would definitely be case by case and tailored by a particular collaborator. And just maybe to finalize, as I talked about, technical expertise and technical excellence and continued development of tools of CRISPR-Cas, that's great. That's certainly only one of the pillars. Regulatory policies, appropriate science-based, that's another big piece that supports technical innovation. Social license or public acceptance, it does impact regulatory policies and public perspective, right? Collaboration, that we also feel that will contribute not only for the technical excellence, but for regulatory policies and social license as well. So these are very, very interrelated components of any innovation. And this is kind of behind our company's guiding principles, how we want to view and how we want to be open and transparent with this technology. And the final sort of little marketing pitch, or for your information, we also have a CRISPR-Cas website at Pioneer, which contains Q&As, it contains Pioneer publications, and this is not tailored to the scientific audience. That website is more tailored to consumers. But if you talk to your families, someone else who might be interested in what they've heard about CRISPR-Cas, and this is a useful resource for information for you. So with that, thank you very much for your attention.
Thank you very much. It was a very interesting talk. Due to time constraints, we can open the floor to one question, or we could hold them to the discussion panel, because I think you led right into where we're going to be at the discussion. But if someone has a burning question, please speak up. Otherwise, we'll hold them to the discussion panel. Sure. Thank you again. We'd like to invite you upstairs for a coffee snack break, and at this time we'll also have our first poster session, which will all be held in the Heritage Room. So if you're a poster presenter, please go upstairs and present your work, and we have 4 judges that will be coming around and evaluating you. And we welcome you all to check out the posters too, because there's some great work up there. Thank you again to our morning speakers.